Hello again. So today we are going to talk about another common molecular biology technique called Immunoblot. And we're going to talk about all kinds of different things associated. So first of all, let's talk about what is an immunoassay exactly. Immunoassay is any biochemical test that's going to help us measure the presence or absence or concentration of a particular molecule in a solution using antibody or an immunoglobulin as our detection method. And these are going to be usually specific against that particular antigen that we are interested in. It relies on the ability of that particular specific antibody to recognize and bind to our specific macromolecule of interest in however complex mixture you have. It could be a purified mixture, it could be a very complex mixture, it doesn't matter. It should be able to bind specifically to your molecule and detect it. For immunoassays, this macromolecule is usually a protein, and any immunoassay that we use is going to be called an analyte. The macromolecule, that is, the protein itself. It comes in two different ways that you can perform these types of assays. Some of them are separation or heterogeneous immunoassays, where you are, separation is where you're separating it out and then examining it. And in heterogeneous immunoassays, you may be looking for multiple things in a separated mixture or in a complex mixture. And these can be both qualitative or quantitative. Some of the methods are just semi-quantitative, some of them are more analytically quantitative and give you a very clear measurable response. So there are several different types of immunoassays that can be performed. Some of them are blot methods and some of them do not require any type of separation or blotting as part of their procedure. The four different ones that I'm going to quickly talk about are dot blot, immunohistochemistry, Immunoblot, and ELISAs. Dot blot and immunoblots take samples that have been extracted, and they are either blotted, dot blotted, just individually. The complex mixtures are blotted onto a little filter, onto a little nitrocellulose membrane or PVDF membrane. In a Western blot, you separate them first with STS page and then transfer them to the membrane. And then we use antibodies specific to our antigen of interest, specific to our macromolecule of interest, and see which areas light up. If you have no signal, that obviously means it doesn't exist. If you have low signal, faint signal, that means it's lower concentration than if you have higher, darker band or signal. So this is semi-quantitative at best, unless you have you utilize a follow-up method, usually computer programming, to examine the relative amount or to quantify the exact amount using a standard control. Then in immunohistochemistry, we use just our tissue sections, so you're not separating anything out of its normal native environment. You're examining it exactly where it is, and you're looking for where that particular molecule of interest is expressed, and then, so its localization and also its general concentration. But again, it's going to be semi-quantitative. And then you have ELISAs, where you use a standard curve of sorts using purified protein or purified macromolecule of your interest, and you compare it against your unknown samples, and you are able to actually quantitate, in many cases, the amount of sample that you have. We are specifically going to be talking about immunoblotting because that's the technique that we are going to be using after we have run our samples, our protein, extracted proteins on STS page. So in this case, our protein of interest is detected either with a polyclonal or monoclonal antibody, depending on our needs, after it has been transferred to nitrocellulose membrane following STS page. So if you remember, last time we talked about STS page and how it separates the proteins by their molecular weight alone, and then we can transfer those separated protein onto a membrane and then detect using the antibodies. So after incubation with the antibody, the protein bands are going to be visualized using a secondary antibody to enhance the signal that is also conjugated to some detection agent so that we can develop it and look at that signal that is produced by those proteins. So this type of technique is what we call Western blotting. The Western blotting technique is based on principles of immunochromatography. So again, we are using a little bit of the same types of methods that we talked about in chromatography. The proteins are separated in the polyacrylamide gel. They can be separated by isoelectric point as well if we did not use SDS and instead used a pH gradient. But they can be also looked at by molecular weight alone if we do use SDS, or we can do what we call two-dimensional gel electrophoresis and separate them by both and get a very unique fingerprint of our individual sample. Once they have been separated, we specifically look for our protein of interest using labeled antibodies. And like I said, it's going to be a semi-quantitative method. It's going to tell you relative amounts in your individual samples and not necessarily exact quantitative amount. You can guesstimate it and use some standards to get that to that point, but it's not going to be 100% accurate. To prepare our samples for that, we prepare our protein extract just like we always do. We can take them from any type of biological tissue, from cell culture, from bacterial culture, even environmental samples. We have to use our lysis buffer based on the type of protein that we are examining and how much of that protein is present, where it is present. So we want to obviously choose a lysis buffer that's going to maximize the extraction of that particular protein. And then based on what it is that we want to do with it down the line, if we are interested in preserving its activity or preserving its whatever post-translational modifications like glycosylations or phosphorylations are present, so that we can monitor their change in different conditions or under different time points or different drug uses, then we would want to add phosphatase and glycosylation inhibitors to our lysis buffer as well to preserve the quality of our protein. We will always add the protease inhibitors to make sure our proteins don't get degraded. And then usually this is going to be followed by what we call quantification and equalization of our proteins. For Western blotting specifically, not only is it important to quantify our proteins initially, but also it is really, really helpful if we go through that extra step where we equalize all our samples that have to be run together for the best results. What that step means is that we want to make sure that all our samples are have the same exact amount of proteins getting loaded onto the gel. This can be done through two different methods. One way is to quantify our proteins and then look at how much concentration of each of those proteins is present, how much concentration of protein is present in each one of our samples. Once we have figured that out, we use the amount of volume that we want to load on the gel, and we say, okay, we want to load 10 microgram of protein. So for my sample A, if I have 2 milligrams or 2 micrograms of protein per microliter, out of those 10, 5 of those are going to be my protein sample. On the other hand, if my second sample has 5 microgram of protein per microliter, I'm only going to need 2 microgram of that protein extract instead. So obviously that's one way that we can load our samples, that we can take into account what is our starting concentration in each one of the samples and make sure that we are taking the exact volume that is going to give us the amount that we need. So 10 microgram or 20 microgram, which is the typical amount that we load in a Western gel per sample. Another way to do that is to prepare, once we have all the quantifications in the first run, we prepare a second diluted amount of our protein extract so that all our samples are exact same concentration. So regardless of whether we started out with a sample that was 5 or 10 microgram per microliter, we change all of them to 5 microgram per microliter. The advantage of that second method is that now I don't have to have a paper in front of me to slow me down and to think about how much volume I need to add for each sample. For all my samples, I need to add the exact volume. So I can just heat them and go to the next step very, very quickly, which is extremely important for good resolution. So I always like to have students do the quantification and then equalize their samples so they're running their entire experimental set at the same concentration so they can get the same volume. This always has to be 10 microliter or 20 microliter, whatever it is, and go on with later life very, very quickly and easily. The second part of this is to choose the right gel for electrophoresis. We talked about this last time when we were just talking about running the STS gel, STS page, and we were thinking about, okay, so if we have larger proteins, we want to run a lower concentration. If we have smaller proteins, we want to run a higher concentration of polyacrylamide. Well, in addition to that, it also matters, usually in an experiment, we don't just look at one protein, we look at multiple proteins. And so we want to think about, okay, are all the proteins going to be within 75 and 37 molecular weight, kilodalton? Or am I going to have some proteins that are really high up and I want to have resolve that as well? And you can look at, many companies will have charts available online to show how different percentage of cells are going to resolve your samples based on some standards. And you can examine those and you can choose the one that's going to separate all your proteins sufficiently. For example, if you have 3 proteins that you have to detect and they're all between 37 and 50, you want to choose a gel that's going to resolve that the best as possible so that, so for example, 12 versus 10%, the 10% gel is giving you more space. The 7.5% is giving you even more space. So that area between 37 and 75 is really, really long in that, so you can easily resolve each one of those 3 individual proteins and examine them very easily. Versus if instead you chose to use a 12%, just, a little bit higher concentration, now that area is really squished up and it may be hard to detect the different proteins that you're trying to look at. So you want to look at these kind of charts very carefully and choose the appropriate percentage for your needs. A lot of times 10%, 12% work the best for most proteins, but that's, I have proteins that I look at, like the histone proteins that are really, really small. So I don't want to, I want to make sure that I'm going to get those as well. And so I don't want to lose them at the bottom of the gel. And I want to make sure that maybe I go up to 15% if I can, so I can detect that area very easily. So that's something that you would always want to think about. Once you know which concentration gel you're going to run, the second thing to look at is the right buffer for your electrophoresis. So you have the typical running buffer that has the STS in it to put all the negative charge and the basic requirements, but nowadays there are multiple formulations of those buffers as well that can again help you resolve different areas of your proteins differently. So for example, in this particular image, you have the same proteins that are run, and they are run using different types of buffers. So in lane 2, you have MUPS, in lane 3, you have the MES, and in lane 1, you have just a typical running buffer. And you can see how the same bands show up differently in these particular buffers. Not only that, if you compare MUPS and MES specifically, again, thinking about those smaller proteins, if you have small, tiny proteins, MES is going to resolve that small protein region a lot better, a lot more clearer than if you used MUPS or just the typical buffer. So that's again something to keep in mind when you are deciding which exact buffer you need to use. MUPS has a higher pH than MES. MES runs a lot faster, but MUPS resolves the mid-ranged proteins a little bit better. MES is going to run those smaller proteins specifically a little bit better. So those are important decisions that we need to make in order for our samples to be best resolved. Because if you have a nicely resolved gel, a nicely resolved and perfectly equalized sample gel, you can get a lot more information out of it and your Westons are going to look a lot sharper and cleaner and ready for the next step. Then we're going to do what we did before. We're going to denature our samples using that STS loading buffer, heating it up to break them all down, make them all linearized, and then you're going to be loading them onto your gels and running them on the electric current. Again, we initially like to run it a little bit faster to kind of stack them in the stacking gel really nice and crisp, and then lower the voltage a little bit so it can resolve slowly and cleanly and crisply. You don't want to go too slow, you don't want to go too fast. If you go too fast, you can get streaking and you can get the gel not looking as crisp and clean as possible. Similarly, if you go too low, it's going to be too slow, and then it's going to give you some loss of proteins, and the bands are going to be fuzzy and not looking very clean as well. Once the gels are done, now we get to the second part, which is to transfer our proteins from the gel onto that membrane. There are two different methods that people use. There are the tank transfer system and a semi-dry transfer system. I'm going to show you examples of both, and then I'm also going to show you what is different maybe in the setup. The biggest difference in the semi-dry transfer is the minimal requirement of the buffer. There's very little buffer needed in there. The tank transfer system is going to use a lot more buffer in the process. In both cases, you're going to start off with your transfer apparatus. You'll have your cathode, the negative end, the anode, the positive end. Obviously, your gel is going to start off on the negative end because everything in there is negatively charged, and then your membrane is going to go towards the positive side. So we usually start with some type of fiber pad, a sponge on our transfer apparatus first. We then place a moist filter pad on top that we then place our membrane, our gel on top of. That gel is then going to be directly in contact with our membrane. At each step, we take away any bubbles. We make sure that the gel and the membrane contact is complete and clean. Then we place another filter pad on top, and then another sponge on top before we close it up and make it into a kind of sandwich. Similarly, in your semi-dry transfer, it's the same idea. You're going to have your filter pads, you're going to have your gel, you're going to have membrane, you're going to have your filter pads, and then you'll have your anode. And in the semi-dry transfer, you'll already have some of those paddings available inside it. These cassettes, once they're prepared, are going to be then added either to in the wet transfer, you're going to add them to your tank of buffer. In your semi-dry transfer, you're going to add it to your apparatus, and then obviously run your current so that your proteins can move from the negative to that membrane and get attached to it. They will attach there, and you'll be able to look at that. Now, what is that membrane made of? Well, again, you have some choices that you can make. There are two different choices, popular choices for the membranes for protein transfer. One is the nitrocellulose membrane, and the other is the PVDF membrane. The biggest thing about these two, both of them work well, both of them work great. Nitrocellulose is going to be a little bit more fragile, so it's going to you're not going to be able to reprobe it too many times, and you won't be able to store it for longer periods of time. However, it gives you really good quality results. Another good part about nitrocellulose, especially when you're learning the technique and starting out, is that you can stain it with Ponzo and make sure that your transfer was good quality and complete. There were no issues. So you can visually see the results of your transfer before you probe it. PVDF cannot be looked at. You just kind of have to believe it until you run the Weston and then find out if it was good or not. The nitrocellulose has a little bit lower binding capacity, so it only takes about 80 to 100 micrograms of protein per centimeter square. PVDF can take twice as much protein, so if you have proteins that are low abundant and you're afraid that you may not be able to get them otherwise, PVDF might be a better size or better membrane to use. Pore size-wise, both of them come in 0.2 and 0.45 micrometer pore size, so you can use whichever one you need. For most of our proteins, we just use 0.45 micrometer. Frankly, there have been very few times that I've had to use 0.2. And like I said, nitrocellulose is a little bit fragile. PVDF is much stronger, harder to cut, harder to play with in some ways, but because it is stronger, it can be stripped and reprobed many, many times. You can even just seal it and store it in your fridge even for 2 years, and then take it back out to reprobe it. If you have a perfect blot, you can reprobe it even a couple of years later with a new antibody or another thing that you need, and get those extra results without having to go through the entire experiment again, which can be a very, very good thing when you're looking at publications and sending papers out and answering reviewer response. The nitrocellulose is well suited for chemiluminescence and fluorescence. PVDF can also be used for most of these things, except that it does give you a little bit high background due to autofluorescence if you are doing a fluorescent Western. So if your antibody can only be detected fluorescently, it may not be a good idea to use a PVDF membrane. For the nitrocellulose membrane, in order for transfer to happen, you have to have methanol in the transfer buffer. You actually use 10% methanol in that transfer buffer. For PVDF, you have to pre-soak the membrane itself in methanol, but you do not need it in the transfer. So it reduces the amount of methanol that you're exposing yourself to that you are having to use. So that's a good thing in some cases. There is a source that I've listed down here that's great for examining things further and learning more about it. So we talked about how transfer check can be a good thing, a positive thing for nitrocellulose membrane. That's what it looks like when we stain with Ponceau. Right here is a perfect blot with equal amount of protein in all the 6 samples. You have your standards on both sides that clearly show it's nice and crisp, everything is straight, there is no curve, there is no issue, there are no bubbles. Here in the second one, you see a membrane that overall was okay. This one they overloaded, but also during the transfer, you see where they did not have a good application of the membrane to the gel, they ended up with these bubbles in the transfer. So imagine a protein, your favorite protein, happened to lie right here. Well, you can detect it very well in this first sample. So unless this is all you needed and this was just a random lane, you would need to rerun that entire gel in order to get that particular protein to be detected appropriately. Also, this particular sample was overloaded because it's higher concentration, right? You can see that this looks different than the others. Frankly, these two look underloaded a little bit now that I look at it. So there are a lot of little issues. Not the worst, but not the best. Here's another one. This looks frankly like somewhere where maybe they were doing an immunoprecipitation experiment. So maybe these two were their extracts in whole, these were maybe their purified ones, and then after another round of purification, and this is the protein they're looking at. Who knows? But obviously, again, you can see the quality and the amount of proteins. Typically, if you're comparing in Westerns, you want all your samples to have, your actual sample pairs. In this case, you would want to have exact same amount of starting material. That's why the equalization step is important. So again, I can see a little bit overloaded in here, though not as bad as right here. So this is where that transfer check with Ponceau is great. And it only takes a couple of minutes. You stain it, and you can destain it in just a few quick washes. So within 5 minutes, you've checked it, and you've moved on to the next step. Now, here again, just to examine the effect of your membrane, which membrane you choose on your protein transfer. Some proteins are going to be better detected with one membrane or the other. So here you can see an example of that, where you had the PVDF membrane at 0.2 and then PVDF and nitrocellulose at 0.45. And for this particular protein, you can see how, the PVDF detected it okay at 0.2, but it was a lot clearer. The bands were a lot sharper, a lot more abundant when they used the 0.45, so it had a better binding with that. And the nitrocellulose really did not give very clean results, same results, but not as crisp. So definitely, if we were wanting to publish this result, we would want to use the PVDF in this case. Here again, you see the opposite result. In this case, Because the PVDF had that stronger binding, you saw that overexposure very easily. So if you have a really abundant protein that's a lot, it's going to be present a lot in your sample, well, maybe you don't want to use PVDF because you'll run the risk of overexposing your sample and not being able to get a good signal. Because if there is too much of that protein bound there, you will not be able to tell if there was too big or too little amount of protein present in your different samples. Here in the nitrocellulose, you get a lot cleaner, a lot crisper results, and you can clearly tell that in this MEL28, this particular protein is present more than in the SY5Y cells. So this gives you a lot cleaner, a lot crisper results, better for publication. Obviously, this would be an overexposed plot that you would not be able to publish with. So that's looking at membrane. Now, once we get the membrane off there, we've checked it. The next step is blocking your membrane. In the blocking step, this is an extremely important step, again, in the Western blots. Without it, you're going to get non-specific binding of all kinds against all kinds of those IgGs that are present in your samples. It prevents our antibodies from just binding non-specifically and gives you a nice, clean signal. Blocking can be done with either BSA or non-fat milk. Typically, non-fat milk works the best, so typically we just use about 5% non-fat dried milk diluted in TBS tween, and we just kind of soak our membrane in it and put it on the rocker for 10-15 minutes at the least. You can put it in overnight, but the least amount that you want to use is at least 15 minutes. And you rock it gently, and it's going to bind to all the non-specific sites. It's just going to kind of make coating all over it, and that is going to help reduce the background. Sometimes you have to play around with the blocking buffers as well. You can, you try non-fat milk, and like I said, 90% of the time it works great. Sometimes it doesn't. If it doesn't, then we try BSA, and if one doesn't work, typically the other one works. And so you can play around with it when you're first trying out a new antibody and see which one is the best. Also, non-fat dried milk is really inexpensive, easy to find. I could just go and pick some up from Walmart for all I care. It's the same one. We just dilute it into the S3. So here to remind you of the workflow, you have separated, you extracted your protein, quantified them, equalized them. You ran them to on the STS page to separate your proteins. You then transfer them to either the nitrocellulose or the PVDF membrane via the electroblot. Then you block it. Once you've blocked it, then you're ready to actually detect your own proteins of interest using your specific antibodies. There are going to be two different steps for antibody. You're going to add a primary antibody. This is going to be specific to your protein of interest. And again, if you remember from the antibody lecture, we talked about how antibodies are made commercially in animals. We use animals for our factories. So you want to know which animal that protein, that antibody was made in. There are many reasons for it. One being, if I have mouse tissue that I'm looking at mouse protein extracted from mouse, I do not maybe want to use a antibody that was made in mouse because it's going to have too much background. All those IgGs are going to non-specifically bind to other places because there's going to be just too much similarity there. You want to typically use an antibody that's going to be specific to your protein of interest from the animal that you got, but not made in that animal. So I may want to use rat one, or I may want to use, for human samples, I may use mouse one or a rabbit one, whatever. But you want to use your primary antibody, and you want to know where it was made. It's going to be important, one, to decide if it's a good one to use, but more importantly, it's going to be important to go to the next step when you are enhancing the signal and you are getting ready for detection. The secondary antibody that we use in a Western is going to be to those IgGs on your antibody that are going to come from the host animal. So if I'm using an anti-mouse primary antibody, then I'm going to use my secondary has to be anti-mouse, right? So if I'm using, anti-galectin 3 made in mouse, then I'm using anti-mouse secondary. If I'm using galectin and galectin-3 antibody made in rabbit, then I'm using anti-rabbit secondary. So my secondary antibody choice is dependent on what primary antibody I'm using. What's the host animal for that primary antibody? And that's going to help enhance my signal, and usually that secondary antibody is linked with some detection molecule that we can then add our substrate to look at the product for. So again, just to remind you, you have your antibodies have some constant regions and some variable regions. These variable regions on the N-terminal are where your amino acid sequences are different to bind to your specific epitope in your protein of interest, and that's what we care about. And obviously, the constant region is the same amino acid in all of them, and that's going to allow us to use that secondary for the enhancement of signal. So our primary antibody is going to be targeting the antigen of interest, so it's against the protein that you want to look for. It's typically very expensive because it's made with that protein in mind, right? So it's made to a very specific part of that protein, or at least it is made specifically to that protein. So that's our, in this case, that's our protein of interest. Our primary antibody is going to bind to that. Now, sometimes that particular antibody can be also linked with some detection method, and you can look at it. If your protein is abundant, maybe that's something you could do. But most proteins will require that secondary step so that you can enhance the signal. So if you were looking at just the primary antibody being detected, then remember, one protein, one antibody. It's going to be a 1-to-1 ratio. Those proteins that are low abundance, you're not going to get very much signal there, are you? So by using the secondary step, which is going to target that constant region of your primary antibodies, much less expensive, you can add whatever fluorophore, whatever color detection method, HR, horseradish, peroxidase, whatever detection method you want onto it, that is going to bind to all the constant parts of it, right? It's just kind of a polyclonal mix that way. So it's going to bind everywhere on that constant region. You're going to get enhancement of signal. So now instead of just having one primary antibody attached, you have multiple molecules attached to it. It's going to enhance the signal that you get from it. So at the end of the day, you have your proteins, you have your primary antibody that's bound to it, you have your secondary molecules. There are going to be multiple ones attached to it. I just showed one in this particular picture. That's going to be attached to some kind of a detection molecule. In this case, it could be peroxidase or alkaline phosphatase. Those are two common ones. It could, then we add our colored product to get, our substrate to get our colored product, which could be fluorescent, which could be, color change, which could be chemiluminescence, depending on what it is that we're using. So Western interpretation, once we have it, it's going to give you a relative protein expression across your different cell cultures or tissue types. We can also utilize it to look for protein localization over time or condition or in response to treatment by fractionating our cells and running the different parts separately. We can also look at it for relative protein levels or activation under a specific condition or with or without treatment. And then we can examine protein-protein interactions using co-IP or co-immunoprecipitation experiments as well. So there are lots of things you can do with the Westerns. I'm going to give you some of these examples. So generally speaking, in a Western, you're going to have your proteins of interest that you run in your different samples, and then you're going to have some type of a loading control. Very important to have that loading control. This loading control is needed to make sure that all your samples were equal. Again, you want to make sure that all your proteins extracts were loaded at the same amount. Same volume, same amount. That's what's important. Loading control is usually a housekeeping protein that's going to be present in all the cells at the same level no matter what. So actin is a very good one, right? Because it's present in all the cells. Structurally, so you'll have abundant amount of it, you'll get the same signal all across as long as you load it the same amount. And then you can use that to compare your own samples with it. In this case, we also have a experimental system check. So here you had just a non-specific siRNA and an siRNA against a specific protein, SIRT1. So we are running antibody against SIRT1 to make sure that our experiment worked. I used an siRNA, silencing RNA, against SIRT1. There should be less SIRT1 in those cell extracts that had siRNA transfected in them. Yes, there is more here, there's less here. Great, my experiment worked. That's important. And then I'm looking at the response of my experiment. So I'm looking at the response of downregulating, reducing the amount of SIRT1 on downstream proteins to see what effect it has. So I'm looking at a protein that is downstream of SIRT1, that's affected by it, P65, and I'm looking at it by looking at its activation, because SIRT1 is activating that protein by acetylating or phosphorylating it, right? In this case, acetylating it. And so I see that under normal condition, you have, when there's SIRT1 there, you have same amount of P65 in all of these three samples, and you have some level of acetyl P65, the acetylated form of this protein. And then once siRNA is done, you have less SIRT1. You still have the same amount of total P65, so it didn't change the amount of P65, but not having SIRT1 increased the amount of acetyl P65. So having SIRT1 was somehow inhibiting the acetylation of P65. That's something that it was doing. That's the response that I'm finding. Here's another one, another example of interpretation. In this case, again, I'm just looking at relative protein amounts. And in this case, I'm using ImageJ analysis or some other imaging software analysis to quantify that response by using my GapTH, my loading control, as my what I'm comparing to. So GapTH showed that most of them have the same amount. This one had a little bit less, so that's going to be taken into account when we do densitometry analysis. And then we are looking at total AKT in the various conditions, and then phosphorylated AKT. The total AKT remains the same, consistent in all of them, regardless of what changes we make, but the phosphorylated AKT is different in different samples for whatever reason, right? So in some cases, you're seeing a difference in those levels. Once we quantify using image analysis, we are able to see that difference a lot more clearly. So most of them, it was, if it was activated, it was activated. But at some point, some condition made a difference. And that's important to note. We wouldn't notice that just by looking at that plot alone without the help of image analysis softwares. Here is a last one that I'm going to show, and this is looking at protein-protein interactions. This is combining co-immunoprecipitation technology with Western interpretation or Western blot analysis. So here I'm examining which proteins interact with my maybe favorite proteins, MRE1 or MRE11. So in this case, we are looking at our full extract, and then we are using flag-labeled Nibrin or flag-labeled XRS2, two proteins that we thought might be interacting with it. The flag label is going to allow us to immunoprecipitate it out without having the Nibrin antibody, for example. So we use the anti-flag antibody to pull out anything that is flag-tagged. And so our flag-tagged were Nibrin and XRS2. So when I use antibody for flag, both of those will light up. We will have XRS2 and Nibrin show up, showing that both of them were indeed flag-tagged, and we were able to see them. So we see our two proteins, and we'll know which one is which based on the molecular weight. Then so these were two proteins that we thought might interact with MRE11. Then we do an MRE11 blot to examine which one of these flagged proteins were able to actually also pull out MRE11. And only flag Nibrin extract, right? So the cell extract that had flag Nibrin is the one that also pulled out MRE11. The cell extract that only had flag XRS2 did not pull out MRE11. When we look at the total lysate, so this is all the cell lysate without doing any co-immunoprecipitation, all of them had same amount of MRE11. So in this case, this is our, in a way, loading control. All of them started out with the same amount of MRE11, but only Nibrin immunoprecipitation led to pulling down of MRE11 as well, not XRS2. And then we look at another protein that might be interacting with them and see that difference as well. Finally, let's talk about some troubleshooting. Obviously, the biggest one would be background issues or loading control issues, right? So you want to make sure your proteins are very well equalized initially. You can even run a gel to check that first before you go through all the trouble of transfer and everything. Background issues could be from buffers that are ugly, that have been reused too many times, not made appropriately, or could be because they didn't block appropriately. Maybe you didn't put enough volume of the blocking buffer in your sample. Protein transfer issues, again, would be when your gel and membranes were put together in the transfer. If you don't take away all the bubbles, roll out all the bubbles, make sure that contact is complete, you're going to get these transfer issues with the bubbles, and that will obviously lead to issues with detection. Also, sometimes you have just too much protein of the one that you are interested in, and you have signal saturation issues. Sometimes you can fix that by doing a lower amount of signal detection. Instead of 1 minute, you do 1 second or 30 seconds. Otherwise, sometimes you just have to reload the entire gel at a lower volume, or lower concentration. So instead of 10 microgram, only load 5 micrograms. So you can see your protein and its individual change effectively. Non-specific bands, again, would be a lot of times because of blocking, but also could be because you use the wrong secondary or the wrong primary antibody. So you have a lot of background issues because of that. And then finally, you could have weak signal detection. Again, many times it would be because of issues with not having the right specific primary antibody or using the incorrect secondary antibody. So many times that if you don't look at the host correctly and use the wrong secondary, of course you're going to have an issue. So just to recap, you are going to be blocking your nitrocellulose. Once you do the transfer, you're going to do a Ponzo test, remove that Ponzo stain, and then you're going to block your nitrocellulose membranes in a solution containing milk proteins. We call it blotto for 1 hour. Like I said, the minimum that we want is 15 minutes. Anything more, that's fine. And then we are going to add our primary antibody specific for the protein that you want to detect in blotto. You want to keep that for at least an hour typically. Sometimes 30 minutes is enough if it's a very good specific antibody. But it is better if you use at least 60 minutes. I usually put my primary antibodies either for 2 hours or overnight for best detection. Then we're going to wash away the unbound antibody before proceeding to the second step. In our class setting with our 4 hours, we do everything together. So we'll already have our membranes in our blocking solution when you come in. And you're going to go ahead and add your primary antibody. You're going to go ahead and then leave that on for an hour to hour and a half before we proceed to the second step. So we'll do 1 hour primary, wash away our antibody that's not bound, and then do 30 to 45 minutes of secondary, wash away unbound antibody before proceeding. For secondary antibody, you never want to go above an hour. You will get a lot of background if you do that. 30 minutes is the minimum you would do. I typically do 45 minutes, maximum an hour. Primary, you can, like I said, leave overnight. This one, you do not want to do that. And then we're going to add our substrate and allow the signal to develop that you can then examine. So hopefully this was helpful in understanding what exactly immunoblotting is done, how it is done, and then some troubleshooting steps as well.